So it's a particular pleasure for me to introduce our next speaker, and that's Professor Michael Laflamme. Michael is the Robert McEwen Chair in Cardiac Regenerative Medicine that we were so fortunate to recruit from Seattle to Toronto, where he's a senior scientist at the Toronto General Hospital Research Institute, and his office is right next door to mine. So I can bother him anytime I wish, which I do, and I thank him for that patience. He's going to tell us today about his really, truly pioneering work in heart regeneration with pluripotent stem cells. Michael.
So good morning. Let me begin by thanking Janet and the Foundation for the invitation. It's a real privilege and an honor to be part of this symposium today. And it's a treat to share with you some of the work we've been doing over the last 15 years or so to try to achieve true heart regeneration using cardiomyocytes we can derive from human pluripotent stem cells. Quick disclosure slide. So I always like to begin my talks with this next set of images that actually comes from the other half of my day job as a diagnostic cardiovascular pathologist. And I like these images because I think they nicely underscore the disease process and what we're trying to overcome. So probably everybody in this room knows what happens during a myocardial infarction. You have rupture of a vulnerable atherosclerotic plaque in one of your major coronary arteries, and that can produce an acute occlusive thrombus. And if that thrombus results in an interruption of blood supply for a sufficient length of time, all the muscle downstream of that blockage is susceptible to ischemic necrosis or infarction. A clinical example of which we see in this specimen was actually obtained at autopsy from an unfortunate individual who died after suffering a large anteroseptal infarct. Now, the good news is with improvements in emergency response times and modern medical management, we're much better at getting folks past the acute phase of an event like this, but then we run into a new problem. That is, the heart is probably the least regenerative organ in the body, and so over time, the muscle that's damaged by infarction is replaced by non-contractile scar tissue. That puts the heart at tremendous mechanical disadvantage, and over time can lead to the development of progressive heart failure. Now, clinically, at present, our only option to replace the muscle that's lost in an event like this is to give somebody an entirely new heart. And as you all know, there's nowhere near enough suitable donor hearts to meet demand. And that's what's driven interest in recent years in alternative cell-based approaches by which we might be able to achieve some degree of cardiac repair. I don't have time to go through all the work that's been done in the field in the last 15 years or so. Suffice it to say, a variety of different cell types have been explored, most extensively cells that we can isolate from the bone marrow compartment, mesenchymal stem cells, and putative resident cardiac progenitor cells. And we're now at a point where about 3,000 patients have received cells like this in randomized controlled clinical trials. And we're able to start drawing some conclusions. So in these studies, it appears that these cells have modest, if any, effects on infarct size. And it's controversial, but there's little or no improvements in systolic function. I would submit one of the reasons for that may be that the adult stem cell types that have been used in these studies show minimal capacity for long-term engraftment and little or no ability to differentiate into cardiomyocytes. Indeed, there's a consensus in the field that to the extent these cells are operational, they're operational through indirect paracrine mechanisms of action. For example, the secretion of proangiogenic factors. Now, that may be very useful, but we should keep in mind that at its core, the fundamental problem in post-MI heart failure is a deficiency of functional cardiomyocytes. What was once muscle is now scar tissue. And with that in mind, our team has focused historically on pluripotent stem cells because this is the one cell type, probably the only cell type, that demonstrates both stable engraftment, as I'll show you, and the capacity to differentiate efficiently into phenotypically unambiguous cardiomyocytes. So our group reported over a decade ago the first protocol by which we could differentiate these cells into cardiomyocytes using entirely defined media conditions. In this particular protocol, we took human embryonic stem cells, we pulsed them with two growth factors we knew from the embryology literature were involved in normal cardiogenesis, namely activin A and bone morphogenetic protein 4. If we then look at the resultant cell populations 2 or 3 weeks later, we find that a high percentage are positive for expected cardiac markers like sarcomeric actin, cardiac troponin T. In fact, with the very best of preparations, we can get better than 90% plus cardiac troponin T positive myocytes. Now, with those very best of preparations, you don't have to resort to something like flow cytometry to tell that your differentiation process is working, and that's because these cells do what cardiomyocytes should do, and you actually can see the contractile activity rippling across the dish by the naked eye. Now, over the years, our group and others have done a lot of work to exhaustively determine the in vitro phenotype of these cardiomyocytes. I don't have time to go into that data. Suffice it to say that these cells have a phenotype that's very similar to cardiomyocytes in the early developing heart. And so that's an important limitation to keep in mind, is that they do have a very immature phenotype. A second limitation is at least the historically available differentiation protocols, like the protocol I described to you a moment ago, actually generate an admixture of cardiac subtypes. So importantly, probably a plurality of the resultant cells will be ventricular myocytes, but they're also going to include significant numbers of contaminating nodal and atrial cells. And the reasons why that's important, I think, will become clear later on in my talk. So rather than focusing on the in vitro phenotype of these cells, what I'd like to share with you is our experience transplanting these cardiomyocytes in various animal models of myocardial infarction. And that work began over 12 years ago with transplantation studies and proof of concept studies in a rat model of myocardial infarction. And the particular study I'll be telling you about now, we actually took animals induced MI, then intervened 4 days later with the direct intramyocardial injection of 10 million human ES-derived cardiomyocytes. And we found that that intervention resulted in the partial remuscularization of the infarct scar. To illustrate that, if the pointer works here, in the left-hand photo micrograph, this sort of pale blue staining area here is the infarct zone. This would be the chamber. And you can see within the infarct zone, we have islands of human myocardium that are growing. We know that this is myocardium because it's positive for expected cardiac markers like beta myosin heavy chain, here detected in red. And we always rigorously confirm donor origin, in this case by doing in situ hybridization with a human-specific pancentromeric probe. We showed that the transplantation of these cells was accompanied by beneficial effects on contractile function. Here I'm showing you fractional shortening as measured by echocardiography. For those that aren't familiar with it, you can think of fractional shortening as the unidimensional equivalent of ejection fraction, so a measure of global cardiac function. And this is a busy plot, so I'll walk you through it. The important bars are the red ones, which represent the recipients of the human ES-derived cardiomyocytes going from 48 hours prior to transplantation, again, that was also 48 hours post-MI, to 4 weeks post-transplantation. You can see over that interval, there's complete preservation of fractional shortening that's unique to the cardiomyocyte recipients, wasn't seen in any of the 3 control groups in this study. And so we actually had 3 control groups. We had infarcted animals that received a pro-survival cocktail. This was a mixture of cardioprotective factors that we showed helped support the engraftment of the cardiomyocytes. We also had animals that received saline alone and non-cardiac ES derivatives. And in the case of all those 3 control groups, we saw the deterioration that you'd otherwise expect in fractional shortening over time. We also looked at regional wall motion in these animals. The best way to do that is by cardiac MRI. And here again, we saw some restoration of wall thickening that was unique to the cardiomyocyte recipients, wasn't seen in uninfarcted controls. In infarcted controls receiving the various vehicles I just described to you. And so this study was actually the first to provide proof of concept in it. We were able to show that there was stable engraftment in these cardiomyocytes and that the transplantation would mediate beneficial effects. In subsequent years, other investigators have gone on to show that these grafts are actually durable. So if you do this experiment in immunodeficient rats, you can go back a year later, you can find graft myocardium, and that the beneficial effects are also durable. So I think the longest anybody's looked is about 12 weeks post-transplantation. If anything, over time, the difference between controls and cardiomyocyte recipients seems to get better. So it was at this point we decided to ask what we thought would be the next logical question, which is we're forming these grafts, but are these grafts actually able to electromechanically integrate? In other words, are they able to couple and fire in synchrony with host myocardium, as would be necessary for them to be contributing functionally meaningful new force-generating units? And to address that question, we actually invested the effort to develop the guinea pig as what we thought would be a more relevant small animal model with which to conduct these electrophysiologic studies. And we had several reasons for focusing on the guinea pig here. The first consideration had to do with heart rate. So if we look at these cardiomyocytes in the dish, they show spontaneous rates of typically about 50 beats per minute, with the very fastest rated contaminating pacemaker cells going up to about 150 beats per minute. If we do something like put a patch clamp electrode on these cells, or have complete electrical control of them, we could stimulate them and get reliable one-to-one capture at rates up to about 4 hertz or so 240 beats per minute. Now the good news is these numbers are reasonably well matched to those of the adult human, but they really raise doubts in my mind whether these cells could ever keep up with something like the sinus rate of the rat. So the rat is firing at 400 beats per minute plus. Now, guinea pigs are a lot faster rated than you or I, but they're sort of unique among small animals in that they have a sinus rate of about 225 or so, which is just comfortably below the maximal rate at which we knew these cells would respond in the dish. And so we reasoned this might be a better small animal model with which to demonstrate coupling. The other nice thing about the guinea pig is it's a popular small animal model for safety electrocardiographic studies. That's in part because at the single cell level, guinea pig cardiomyocytes have electrophysiologic properties that are much better matched to those of adult human cardiomyocytes. Here I'm just showing you one of those electrophysiologic parameters. This is the action potential duration to 90% of repolarization. And you can see how much closer the numbers are for guinea pig myocytes than our rats relative to humans. And finally, the very practical consideration is over time, after decades of working with rats, I'd become extremely allergic to them. I didn't like the idea of my lab would be working on an animal model that I couldn't even be in the same room with. And so with those considerations in mind, we set up the experiment I'm outlining for you here. So we induced injury again in this guinea pig model. We intervene in this case with 100 million human ES-derived cardiomyocytes, again, directly injected via thoracotomy. And then our endpoints included histology, contractile function by echo, and we also performed an ex vivo imaging technique that I'll describe to you in a moment. So in terms of structural outcomes, they're very similar to what we'd seen previously in the rat model. Again, we were able to get robust remuscularization of the infarct zone. And we saw improvements in indices of contractile function by echocardiography. Here again, I'm showing you fractional shortening. But our focus in this study was to ask the question whether these cells were able to integrate electromechanically. Now, we knew that they had the subcellular machinery that's required for that. We could tell that histologically. So we could look at sections and identify areas of contact in the border zone. And we saw evidence of shared gap junctions between host and graft cardiomyocytes there. We could demonstrate that by immunostaining with an antibody against the dominant cardiac gap junction protein, connexin 43. We also could see elements of the mechanical junctions between host and graft cardiomyocytes by immunostaining with elements of the adherens junction like NK-adherent. But we wanted to get more directly at this question whether these grafts were functionally integrated. And to answer that, we actually engineered human ES lines and later iPS lines that stably expressed a genetically encoded calcium sensor. This is the protein GCAMP3. You can think of GCAMP3 as a fusion protein that includes the calcium binding moieties of calmodulin that's been derivatized with GFP, such that in the presence of elevated calcium, this molecule undergoes a conformational change and an increase in fluorescence. And the long and the short of it is cardiomyocytes that are expressing GCAMP3 show robust fluorescent transients with each contractile cycle. And so the readout here in vivo is relatively straightforward. We can transplant cells expressing GCAMP3. If we see a flash of green light, we know the graft is active, and then we can line that up against the host electrocardiogram to ask whether it's one-to-one coupled. And so it's a very convenient graft-autonomous reporter of graft activation. And so we began with transplantation studies in normal, that is, uninjured guinea pig hearts. And what you're actually seeing here is a montage of grafts in uninjured recipients, here using an open chest preparation. And I think you can see it's sort of bright in the room, but it looks like all regions of the graft are flashing in nice one-to-one synchrony with the QRS complexes of the simultaneously acquired electrocardiogram. Now, one concern you might have about that experiment is about confounding motion artifacts. And to deal with that, we like to take these hearts out of the chest. We can keep them alive ex vivo in something called a Langendorf apparatus. And under those conditions, we can perfuse the heart with mechanical uncouplers, so drugs like blebistatin and BDM, that arrest motion but allow persistence of electrical activity. And under those conditions, we still saw nice one-to-one synchrony between the GCAMP fluorescent transients and the QRS complexes of the host electrocardiogram. And so this was definitive proof that the grafts, at least in uninjured hearts, were able to couple, which is a sine qua non for true heart regeneration that hasn't been shown with any of the other cell types I told you about before. So whenever we've looked after transplanting these cells in uninjured recipients, we've always found one-to-one coupling over 100% of the visible graft footprint. We can pace these hearts up to about 5 hertz or so and still get nice one-to-one capture. If you go much above 5 hertz, the guinea pig heart doesn't really tolerate it very well. And while it's hard to prove a negative, my laboratory has looked at this pretty hard, doing the same sort of experiment in the rat model, and we've never seen what I would consider convincing one-to-one coupling in the rat heart. Now, if you slow a rat heart down to 40, 50 beats per minute, and we could argue about whether it's really still alive at that point, you might see episodic flashes of coupling, but I don't think it's going to be a model that's going to be useful for asking integration in situ. And so for us, that sort of validated the investment we had made in developing this other animal model. So the studies I just told you about came from grafts that were formed in normal hearts. And of course, in that context, you're going to have a lot of contact between host and graft myocardium. It's a very different context than what we're going to be talking about with delivering the cells into scar tissue. So there you're going to have all of this intervening fibrosis that you might expect would act as a barrier to electromechanical integration. And that's indeed what we found to be the case. So here I'm showing you one of our success stories. So in a majority of hearts, we saw what I'll be demonstrating here. So just to orient you, this is a guinea pig heart. Again, we're looking at it from the epicardial surface. The border zone would be running right along here. All of this is scar tissue in sort of the upper right-hand corner. And I've circled two graft regions for you. So one is out here in the scar, and the other one's nestled in the border zone. And if you look carefully, I think you'll see that both graft regions are firing in synchrony with the host electrocardiogram. Unfortunately, that wasn't always the case, and we had a minority of hearts that were like the one I'm showing you here. So if you look carefully in this case, the graft region circled in red is nicely one-to-one coupled, but the one in blue is not. It's essentially marching to its own drummer. So in some of these hearts, we don't just have a problem of host-graft electrical mechanical integration that we need to overcome, but also graft-graft integration. And as you might imagine, this is something that we're very interested in focusing on, and later I'll highlight some of the ways that we might be able to improve this down the road. So we've seen one other example of what I would call bad electrophysiologic behavior by these cells, and that's in a subset of these hearts in which the graft was coupled. We've seen instances where the graft can act as an ectopic pacemaker. And to demonstrate that, we've actually done experiments where we've identified a novel class of fluorescent voltage sensitive dyes that will actually label both host and graft tissue. And so we can image these hearts and interrogate with high spatial and temporal resolution what's going on in both tissue compartments. And in some of these experiments, we've actually used the GCAMP graft autonomous reporter activation to be sure that we're discriminating what's in graft tissue. And I'm going to play you one of these videos. In this case, we've loaded the heart with one of these special dyes, a dye called an EpiQ. And so it's going to be reporting activation, electrical activation in both host and graft tissue. And for the purpose of this experiment, as you'd expect in a normal heart, we're going to see electrical activation starting at the apex of the heart here on the right-hand side, and then proceeding up towards the base of the heart at the left. And I'll play the video, and you also see the simultaneously acquired electrocardiogram. What you're going to see in the first beat is normal activation, then a second electrical systole, normal activation, and then I want you to pay very close attention to what's happening during this blip right here. So normal activation is going to proceed very rapidly from right to left. You're going to see that again here in a second, going from right to left. And then you're going to see what happens in this impulse here. This is what we called a premature ventricular contraction. And if you're watching very carefully, you actually saw that activation began at a different focus there. It was right smack in the middle of the field, which happens to be right smack in the middle of our graft tissue. And I know that went by fast, so I'll show you these are representative activation maps for those individual host and graft-based beats. And so what was happening in this animal is about every fourth or fifth beat was actually arising from the graft tissue. It was breaking through periodically. And it's easy to understand how this could be pro-arrhythmic, and you'll later see why that's of particular concern to us as we move forward in what is a translationally oriented program. So as a translationally oriented program, in the remaining few minutes, I actually want to tell you about some very recent work we've been doing in a pig MI model with, again, the goals of eventually making this into a useful therapy. And I should say up front that this is very new data. This is entirely unpublished work that we've just done in the last about 18 months or so. So bear with me with it. So the rationale for doing transplantation studies in a pig MI model should be pretty obvious. The cardiac anatomy and physiology of the pig heart is almost identical to that of the human heart. One important consideration is the heart rate of a pig heart is pretty much identical to that of a human. And that's particularly relevant if we're worried about the possibility of graft-related arrhythmias, because in a faster rated rodent, it might mask arrhythmias that we would otherwise see in people. The other nice thing about the pig is because of its size, it enables pretty much direct translation of various imaging and catheter and clinical surgical procedures. You can go pretty much from pig to humans. And that's particularly relevant for us because going forward, we actually want to switch from delivery of these cells via an open chest intramyocardial injection to delivery via available catheter-based transendocardial injection catheters, which would move this into the realm of minimally invasive therapies. And it'd be impossible to breach that sort of approach in a small animal. And finally, just a very practical consideration, the pig is widely accepted by regulatory agencies for preclinical safety and efficacy studies. If you have a novel cardiac intervention, you pretty much are expected to show it in the pig before you move into the clinic. Now, the downside, if you will, of a pig, but would be true in humans as well, is we're talking about a much larger dose of cells, right? So in the rats I told you about, we were delivering 10 million cardiomyocytes per heart. In the guinea pigs, we got up to 100 million. In the pilot study I'll tell you about in a moment, in these pigs, we're delivering 1 billion human-derived cardiomyocytes, which happens to be the same number of cardiomyocytes you lose in a typical human infarct. And that's where that number came from. And so to meet this demand, we're taking advantage of the tremendous capabilities for cardiomyocyte manufacturing that have been developed by my collaborators here in Toronto. And this is really work that I should credit to collaborators including Gordon Keller, Peter Zanstra, and scientists at the Center for Commercialization of Regenerative Medicine. And what they've done is they've taken a different differentiation protocol, but one that involves the same growth factors, activin A and BMP4 that I told you about before. But here, instead of doing this in static two-dimensional culture, we're doing it in suspension culture and stir tank bioreactors, so small aggregates of pluripotent stem cells that are then differentiated, again, as small aggregates. And it turns out in one 500 milliliter bioreactor, you can generate on the order of about 10 to the 9th cardiomyocytes, which again is sort of our dose range. And if you look at the resultant cells, again, a high percentage will be positive for markers like cardiac troponin T. In fact, for the transplantation studies I'm about to tell you about in a moment, these had a mean cardiac troponin T purity of about 86%, so reasonably good. And the balance of contaminated cells mostly look like they have a fibroblastic phenotype, at least based on immunophenotyping. And so I'm happy to report to you that again, this is a pilot study that's really just getting underway, but we're able to get pulse remuscularization in the infarcted pig heart with outcomes that look very similar to what we'd seen in the preceding small animal models. And so I'll draw your attention here to the upper photograph. What you're looking at here is a transfer slice, the entirety of the pig heart, and all of these dotted areas you see here is graft myocardium that was growing, at this case, at a 4-week time point in infarcted and immunosuppressed pigs. And this is one of those instances where size matters, so I'll draw your attention to the scale bar here. So this instance, we're actually forming with the delivery of 1 billion cardiomyocytes on the order of cubic centimeters of human myocardium, which we think is the scale of regeneration, the scale of remuscularization that you need to think about to meaningfully impact an infarcted human heart. If we look at higher magnification, as you might expect, the cells are expressing various anticipated cardiac markers. Here I'm showing you alpha-actinin, cadherin, cardiac troponin T, connexins, the like. We actually have underway a dose-response study with these cells. So far, we've only quantitated the animals that have received 1 billion cardiomyocytes, but I can tell you at that dose, we're getting graft sizes that are in the range of about 13 to 20% of the total infarct footprint. We think that's probably not enough, but we're hoping that with either a higher dose of cells or using some interventions that we've previously have shown have enhanced engraftment in small animal models, we might be able to improve this parameter somewhat. Nonetheless, this is, again, a respectable amount of myocardium, and one of the things that's really excited us is it's enough myocardium that we've shown recently that we can actually see this myocardium using standard imaging modalities like cardiac MRI. And so we've actually carefully correlated the histology with the late gadolinium enhancement MRI images. This is a standard technique that we would use in our patients anyways, and we could see a beautiful correspondence between the two. And this opens up the possibility that we could monitor these grafts over time to see how they're doing without the requirement for any kind of exogenous labeling agent or the like. Now, I spent a lot of time talking about the potential pro-arrhythmic behavior, so probably the next set of what I'm going to tell you about won't be a great surprise. It's something that we expected sort of based on first principles and the preceding work on the small animal models, and that is in these pigs we are actually seeing a fair amount of graft-related arrhythmias, with these animals showing frequent bouts of a type of arrhythmia called ventricular tachycardia. Now, all of these animals have been in normal sinus rhythm prior to transplantation. And then about 3 to 4 days post-transplantation, we're seeing the emergence of bouts of initially non-sustained and later sustained ventricular tachycardia. As I'll show you in a second, peaks about a week or two in. But interestingly, it seems to be a transient phenomenon, such that by 3 to 4 weeks post-transplantation, they've all been back in normal sinus rhythm. Here's a little bit of quantitative data. The y-axis is important here. What I'm plotting for you is the percentage of time in each 24-hour monitoring period that these animals were in VT. So it's important to keep in mind that during the peak of arrhythmogenesis, these animals are spending the majority of their day in VT. But for whatever reason, it seems to be dissipating over time, such that by 3 or 4 weeks later, they're back in normal sinus rhythm. We're convinced that this is actually a graft-related phenomenon. So if we look at infarcted animals that have received vehicle control, so far I think we've seen 2 animals that both showed 1 instance of VT each, and those only lasted a few seconds in duration. So these are really being driven by the graft myocardium. Now, as you might imagine, we're very interested in understanding the mechanistic basis of these arrhythmias. We're focused like a laser on that right now. One of the things we're doing is applying some of those optical imaging techniques I described to you before. But one of the really nice things about the pig is there's actually clinical tools that we can bring to bear to investigate the mechanistic basis of these arrhythmias. And one of those tools is a technique, a catheter-based technique called electroanatomic mapping, where we snake a catheter that has an electrode array into the left ventricular chamber, and you can use this to generate highly detailed 3-dimensional maps of electrical activation as it's occurring in situ. And this is something that we've been doing recently in collaboration with a clinical electrophysiologist and scientist at University Health Network, Kumar Nantha Kumar. And so what we've been doing in these experiments, we've actually been mapping the animals during the peak of arrhythmogenesis, so 10 days post-transplantation, and we've been generating activation maps like the one I just described to you. And in contrast to infarcted controls that have received vehicle, we're seeing a very different pattern of activation in these cell recipients. So we're actually seeing what we call focal automaticity, with a focus of earliest activation that's occurring down near the anterolateral apex, which happens to be exactly where we're implanting the cells. So that really looks to be the mechanistic basis of the arrhythmias. We've ruled out, we haven't seen other potential mechanisms like reentrant phenomenon we see no evidence for. And we've actually been able to very carefully register our 3-dimensional histologic data sets with these electroanatomic maps I just described to you. And every instance we've found graft myocardium right at the focus of earliest activation. So it looks like the mechanism of arrhythmogenesis in these animals is the graft acting as an ectopic pacemaker, just like we would have predicted based on what we saw in the rodent studies I told you about earlier. Now, obviously, that's concerning, but to us, in a lot of ways, this was a good news story because this increases the likelihood that this is a graft-autonomous phenomenon. And as I told you, the cells that we're transplanting, we know right now, include an admixture of cardiac subtypes, including a substantial fraction of pacemaker cells. And my colleague Gordon Keller has actually developed refined differentiation protocols that actually result in a homogeneous population of ventricular myocytes that are more electrically quiescent. And so the very next experiment we're going to do is do exactly what we've done here with those ventricular enriched populations and see whether it reduces or eliminates the arrhythmias. Don't have any data to share with you yet. So let me summarize what I told you this morning. So the good news is large-scale remuscularization of the infarct scar is indeed possible using human pluripotent stem cells, including in the case of human-sized infarcts. Obviously, the biggest hurdle we have to deal with is this phenomenon of graft-related arrhythmias. And so we're testing a variety of strategies to try and improve or tame the electrical behavior of these grafts. I told you about one of the strategies, which is to use these ventricular-enriched populations. We're also investigating various scalable ways by which we'll be able to mature these cells in vitro prior to transplantation. The pig study I told you about, none of these animals were treated with any kind of antiarrhythmic drug in the first place, so obviously we need to go back and see if some of the clinically available and even some unconventional antiarrhythmic drugs may improve some of these outcomes. And finally, we're really excited about applying CRISPR technology. So we're pretty sure we know what are the culprit ion channels that are driving these arrhythmias. We can go in and modify them in the cells and see if that changes the outcomes as well. We have a few other challenges before we can move this forward to the clinic. Obviously, one of the things we're very actively working on, in particular with a new commercialization partner, is trying to establish scalable and economically viable cardiomyocyte production, and eventually that'll have to be done at clinical grade, not research grade. We're going to have to deal with the immune system. We have some strategies I didn't have time to go into to prevent graft cell immune rejection. Obviously, if we're talking about end-stage patients, these are the kind of patients that get a heart transplant, so we're comfortable applying some degree of immunosuppression in those patients. And finally, we need to demonstrate efficacy in a large animal model, and that's something that we feel like we're now well set up to do in the pig model, which would really be the best model to ask the question. And so with that, I need to thank the many folks that contributed to these studies. In particular, if I could highlight my long-term collaborator and mentor, Chuck Murray, who did the rodent studies I described with us. If I could also highlight a couple key individuals that contributed to the unpublished pig data I showed you at the end. That was led by a talented postdoc in my lab, Rocco Romagnolo. This has been a real collaborative effort. It's involved scientists including Gordon Keller, Peter Zanstra, the folks at the CCRM I mentioned to you. We're working with Graham Wright and Nilesh Gugra at Sunnybrook, and the mapping studies I told you about were done with Kumar Nantha Kumar. With that, I thank you for your attention, and I don't know if there's time for any questions or not.